is a fascinating topic, and unless you've been hiding under a rock for a few years, this is the big thing right now in almost any discipline in medicine, maybe not every single one, all right? So excited about it. I hope you get something out of it and understand a little bit about how it is having an implication on our behavior cases, and how we might be able to use this even more going forward. So the first thing is, what is the microbiome? And the microbiome, and there are many of them, is the microbiota, and we're going to define that, but the microbiota are the actual organisms. So they're bacteria, archaea, prokaryotes, viruses, fungi. So microbiome is all those organisms and the products that they produce. So these are cellular organisms, and just like any cell, cells produce things and then they dump them out. Like your cells in your pancreas produce insulin and they dump them out, and they produce digestive enzymes and they dump them out into your body. So all these organisms in the microbiome also produce things, and the things that they produce have an influence on the environment around them. So when we say the biome, we not only mean the organisms, but we mean all that stuff that the organisms make, right? Every body surface has a microbiome. You have a microbiome in your mouth, in your nose, in your GI tract, on your eyelids, and your skin, okay? The one in the GI tract is the most diverse and the most complex. And when you really dig into it, I'm telling you, it is super, super complex, which is probably one of the things that fascinates and excites me about it. So the gut microbiome is the largest metabolic organ, right? In the proximal, the stomach and the proximal small intestine, so the duodenum, there's not too many bacteria and microbiota there, but when you get into the distal small intestine, like the ileum, and into the colon, especially in our hindgut fermenting species like horses, rodents, people are kind of hindgut fermenting to some degree, then we're talking about gazillions of organisms. So there are 10 trillion bacteria per mil cc of fluid in the colon. Okay? So it's like our national debt back there. So all of these microbiota release compounds that affect our immune function, the maturation of our brain, our immune system, the intestinal tract, and affect behavior as well. So we're going to go through some of this and some of these compounds in a little more detail as we go through the lecture. So one definition that you're going to come across if you read about this is called dysbiosis. And dysbiosis actually is just an imbalance in the bacterial composition of whatever microbiome you happen to be discussing. So this could be changes in the microbial number, changes in the ratio of the good bacteria to each other, or it could be changes in the ratio of good bacteria to bad bacteria in that particular microbiome. So in veterinary medicine, we have, and in people too, we have this term called SIBO, which is small intestinal bacterial overgrowth, or again, dysbiosis. So they're used interchangeably, and really dysbiosis is probably a more accurate term because bacterial overgrowth implies the problem is that there are too many bacteria, which isn't always the case. Sometimes when we have intestinal tract disease, the problem is that the diversity of the organisms have changed, or again, it's just a shift in the ratio of the different bugs rather than a change in the entire number of the bacteria. So what influences your gut microbiome? Everything. It is a very dynamic entity. It changes over your lifespan. It changes with your diet, which we're going to talk about a little bit more. Your genetics affects it. Your age. So there is a definite change across your lifespan, and this is true in animals too. The microbiome and its characteristics when perinatal or at weaning is not the same as when you're a senior, and that's true again across all species. Your gender or sex affects your microbiome, so sometimes things that happen to us, especially early in our life, have gender differences in how it impacts your physiology, and that's true with the microbiome as well. Your sleep cycle affects the microbiome, so shift workers have abnormal alterations in their microbiome because they're not in a normal day-night cycle. Exercise affects it. Your circadian rhythm. So circadian rhythm is just a 24-hour rhythm. Everybody has a 24-hour rhythm, so to speak, in their body. And what's also fascinating is your microbiome also has a circadian rhythm. About 20% of the bacteria in your microbiome change across a 24-hour period because those cells have their own little circadian rhythm that is again affected by other things like diet. When you eat is actually a large factor influencing the circadian rhythm of your microbiome. So other big ones are location and geography, where you live. So individuals that live on one part of the country or a different continent or wherever are going to have different microbiome from those in another. Your household members and social companions. People that live together have more overlap in their microbiome than people that don't, and related people do. You share a little bit of microbiome with the pets that you live with. Okay? I highlighted these because we're going to talk about these in some more detail, and because they're big ones, and they're ones to some degree that we can actually influence. I can't change my genetics. Is my microphone still working? All right. I can't change my genetics, and I can't change my gender, but I can change my diet and exercise and sleep habits. We're not going to talk about those two as much. And you can change your environmental exposure. So environmental exposure, what I'm talking about, is especially early in life and what they call this hygiene hypothesis. How many of you have heard or read about the hygiene hypothesis? Everybody's actually probably heard about this. This is the idea that in our society nowadays, that because of the use of antibiotics and our obsession with cleanliness and antibacterial things and antiseptic and all these hand sanitizers, that we are actually making ourselves sick. Because when you live in a lower or a more aseptic environment, your body isn't taking in the microbial diversity that it needs when you're young for your immune system to develop tolerance to antigenic diversity. So there is actually data out there that kids grow up on farms and play in horse poop. Yes. Are less likely to develop asthma and allergic disease than those that live in this pristine environments. So I go out to the barn a lot and I come home, you know, and then I get ready to make dinner, and a lot of times my husband's like, "Did you wash your hands?" And I'm like, "Yeah." When secretly I'm going, "Really, I'm doing you a favor." Okay? So the other one, as I said, is diet. We're going to talk about that a little bit more later, so I'm not going to go into much detail now. We're also going to talk a lot about disease and stress. So these are some of the common bacterial phyla in people, dogs, cats, horses. All right? As you get to the end, I don't have too much for you parrot folk, I'm sorry, but I have a couple little statements. So, again, these will come up again. I just wanted to look at, you know, when I was reading through all this, I'm like, oh yeah, you know, the Firmicutes, phyla, and then there's these bacteria, and like, I needed a hierarchy. And so I put the major 4 phyla, and I just put some examples of classes of bacteria under that. And I mostly did that to help keep these straight in my head. But of course, I thought it would be helpful for you too. All right, so first we need an anatomy lesson, because if I'm going to tell you how this works, then we need to know what everything looks like. And somebody was going to see if they, can everybody see these little like yellow bands here? Well, that's what I was trying to get somebody to do. Yeah. I forgot, I didn't see you back there, I'm sorry. So this is a diagram of the lining of your intestinal tract, all right? So this is the lining of the intestine. These are little crypt cells, and usually they're little villi. They're like little fingers that go up also. And there's all these different kinds of cells that line the intestinal tract, okay? So this is the body side, and up here is what we call the luminal side. That's the inside of the tube of the intestinal tract. That's where the food and the doo-doo go, okay? Just so you're oriented. So if you see this little yellow band, can everybody in the back see that? That is the mucus layer that adheres to the endothelial cells. So these are endothelial cells. There are some other cells mixed in there that I'll show you later, all right, the epithelial cells. So in the small intestinal tract, there's one mucus band, and that's where a lot of the bacteria live. The colon has 2 mucus bands. The inside band that sticks tight, and then there's an outer layer of mucus in the colon that's a little looser. And if your colon's healthy, that's where all the bacteria are. If the bacteria get into the inner band in the colon, then you're in a little bit of trouble, okay? So there are 2 basically microbial communities. There are microbes that are loose in the lumen, and then there are microbes that are in this mucosal layer. They are distinct, but there is some overlap between them, and obviously there is some transition between them, but they are distinct and they have different functions, okay? All right, so this is a blow-up of some of these cells right here, okay? So these are what we call enterocytes. And these little fingers, for those of you who remember active physiology, it's called a brush border. And these little fingers have lots of other little fingers on them. And those little fingers have a lot of surface area, because this is where a lot of the nutrients and stuff are absorbed into the cell that pass through then to the bloodstream, okay? Because we kind of have to know how stuff gets from the lumen into the body, all right? So what's important about this slide are these tight junctions right in here. These are tiny little protein bridges that lock the cells together, all right? They keep the cells bound together. So we call these tight junctions, right there, tight junctions. They should be tight. If they're not tight, then you have a problem, all right? So when stuff passes from the lumen into the body, it can go through the intestinal barrier 2 main ways. And one is it can go through the cell. It can be absorbed into the cell. There may be some processing, and then it is passed out into the bloodstream, okay? The second way is what's called paracellular transfer, where it passes between the cells through those tight junctions. So you can already imagine, and we're going to talk about this more later, but you can already imagine that if that little bridge fails and it allows the cells basically to separate, a lot more stuff gets through, and a lot of times it's stuff that we don't want to get through, okay? Everybody with me so far? Yes. Okay. All right, so what does the gut microbiome do? It is involved in every aspect of host physiology. And when we say host, we basically mean, obviously, the individual. Some of these we're, again, going to talk about in more detail, but it is heavily involved in the maturation of your immune system. And I alluded to that already a little bit ago with the hygiene hypothesis. It is very integral for establishing and maintaining the function of the barrier, those tight junctions, okay? It is involved in synthesizing and extracting nutrients. So there are some things that we eat that we can't digest. We don't have the enzymatic processes to digest certain types of fiber and stuff. And so what happens is those foods pass down into your colon, and all those little bacteria sit there, chew that stuff up, turn it into other products that we are then able to use in some useful way, okay? Microglial cells are ancillary support cells in the brain that are involved in protecting the brain from injury. It's basically kind of a type of an immune cell. It's also involved a lot in synaptic pruning. So when synapses sort of degrade, they have to be cleaned up, if you will. And microglial cells are involved in that process. So the gut microbiota is actually involved in the maturation and function of those cells in your brain. And again, we're going to talk a little bit more about how does that message happen, okay? It's involved in regulation of the myelination of the nerves in your prefrontal cortex. So how important is the prefrontal cortex? Pretty important. It's involved in the function and maturation of the blood-brain barrier. It is involved in establishing and influencing our hypothalamic pituitary adrenal axis. This is your stress response system, okay? The microorganisms also, some of them, produce neurotransmitters on their own. So for example, streptococci, enterococcus, Escherichia, so E. coli, it's not actually E. coli per se, but that genus Escherichia, those bacteria produce serotonin in the GI tract. GABA is produced by lactobacillus species. All right, so not all our neurotransmitters actually come from our own body. Some of them actually come from the organisms that are inside our gut. All right, so how do these two communicate? Well, they don't know everything, and I don't understand everything, but I'll tell you what I know so far. This is a bidirectional communication, and information goes both directions, obviously, okay? So here are little enterocytes again. The little finger side of the cells always project towards the lumen, just to help you orient when you see different diagrams in this lecture. So here are all our little microbiota in here, our enterocytes. This little blue cell over here is called an enteroendocrine cell. That cell specifically produces different types of endocrine products and hormones, okay? So it'll come up again in the future. This obviously is not a to scale diagram because your brain doesn't sit, hopefully, quite that close. So there are all different types of neurons that are outside the brain. So these are neurons in your periphery that attach to these enterocytes. So that means anything that happens to these cells can hook to receptors on these neurons, which communicate to the brain, right? So these are the main ways that the gut and the brain communicate to each other. One, we have what's called vagal and spinal afferents. So the vagus nerve is one of the cranial nerves. It's one of the major nerves in your body, and basically if it wasn't there, for the most part, you would die, all right? It's super, super important. And if you cut the vagal nerve in some of these organisms, like these mice that they do a lot of studies on, the transmission between the gut and the brain sometimes then doesn't happen. So it depends on the mechanism, but we know that some of the messages that are coming out of the gut are transmitted through the vagus nerve, because if you cut the vagus nerve, message doesn't go, okay? So it goes the other way too. So that's one mechanism. The other one are these called cytokines. So cytokines are basically kind of protein molecules that cells produce, immune cells produce, that are used to fight or promote inflammation, okay? So any of the cells, so here are some immune cells. These are lymphocytes. Any of these cells that produce cytokines, those cytokines get into the bloodstream. Sometimes the cytokines pass through the cells into the lumen and actually affect the bacteria that are in the lumen of the GI tract. But these are other ways that the message goes. So if these cytokines get into the bloodstream and then they are able to go up to the brain and pass through the brain barrier, that's one way to get a message. But these cytokines also affect other cells and nerves in the body, which then can also communicate to the brain. Okay? Still following me? Yes. Okay. So then one of the other ways are circulating gut hormones. So remember I told you that these little cells produce hormones? So not only do these cells produce hormones in relation to what happens inside the gut, but sometimes these bacteria, again, also produce substances that can get into the bloodstream, either through the cells or between those tight junctions. And sometimes these are substances we want to pass through, and sometimes they're substances we don't want to pass through. But still, once they get into the body and into the bloodstream, they can go affect organs everywhere, and they can affect your physiology and the rest of your body, including in your brain. Okay? So these are our main pathways for communication between the gut and the brain. So as I mentioned before, these little endocrine cells produce over 20 different kinds of gut hormones, okay? And I don't know what they all are, and I can't tell you what they are. But they are messengers between the gut and the brain. And then not only do the microbiota produce their own things, but the other thing they do is they control the body's access to building blocks for other compounds, such as neurotransmitters. So most of our neurotransmitters come from amino acid bases, or some of them do. And if the microbiota, to some degree, control the amount of amino acids that are in your GI tract, then those bacteria can influence the number or the amount of neurotransmitter that is synthesized inside the body. So if some of the bacteria use up tryptophan, for example, and consume a lot of it, then how much tryptophan does our body have to make serotonin? Not so much. If the microbiota do something that actually produces more tryptophan, now our body has the ability to make more serotonin, which then some of that's going to be synthesized inside the brain and potentially affect our behavior. So I also mentioned before that the gut-brain or the microbiota affect the blood-brain barrier permeability. So this is now the blood-brain barrier, although it looks somewhat similar to what you saw in the intestinal tract. So here are the little cells that line the barrier, little membrane that separates. This is a neuron in green, and these are some of the support cells. Remember I mentioned the microglia right there. These are other support cells that help keep neurons functioning and alive, all right? So when substances get in the blood, the cells that line the blood-brain barrier have their own little tight junctions. And if those little tight junctions fail, for a variety of reasons, then we get passes of substances into the brain that, again, shouldn't be there, all right? So similar process to what happens in the intestinal tract in terms of keeping stuff in or out of the important part of the body, which is your brain. I mean, they're all important, but. All right, so let's talk a little bit about how the microbiome develops. There is some data to show that the gut starts to colonize a little bit in utero. So for example, there are some, this is probably mostly in mice, that the meconium, which is sort of the first stool that an animal passes after it's born, there's actually microbial DNA there. And obviously that didn't happen at birth, so there's probably some colonization that happens in utero, but the bulk of it happens at birth, and the form of the birth, C-section, vaginal birth, whatever, actually influences the organisms that start into your GI tract, okay? You get very significant changes at weaning because there's a dietary change, and as I've mentioned before, diet plays a huge role in the diversity and number of microbiota in your tract, right? So one of the ways they study this is they breed what are called germ-free mice. So germ-free mice are basically sterile. They do not have any organisms in their gastrointestinal tract. And then they compare them under different tests to regular mice. And what we know about germ-free mice is, ironically, they show less anxiety than regular mice. However, they have exaggerated HPA axis reactions to stress. So their baseline anxiety is lower, but when you stress them, their body responds in an exaggerated manner, and their the corticosterone is the rat and mouse version of cortisol. So their baseline rat cortisol or mouse cortisol levels basically are higher than normal mice and rats, okay? We also know there's stuff that's happening in the brain, like their hippocampus and in their cortex. There's a compound called brain-derived neurotrophic factor, and it is involved in producing new neurons, but also new synaptic connections. And we know in germ-free mice that BDNF is lower in certain parts of the brain compared to normal mice. So how does stress affect this, right? So here are germ-free mice. These are things that we see behaviorally. Anything in red is lower in germ-free mice, and in green, it's higher. So germ-free mice move more and they self-groom more, but they have lower levels of anxiety, they're less sociable, and there are some detrimental effects on memory formation, okay? They have lower BDNF, they have higher, ironically, serotonin levels, but their hormones related to stress, baseline, are also higher, okay? So there is this altered stress reaction system. If you recolonize mice within 6 weeks of birth, this will normalize. If you do not recolonize them within 6 weeks, but you recolonize them later, they will be basically abnormal forever. So there's this short window where you can try to sort of fix them. Now that doesn't mean the 6-week mark happens for people and dogs and stuff too. We don't have those kind of numbers. Like, we don't know if you take a puppy that's not that there's ever going to be a germ-free puppy or a germ-free kid, you know, we hope. But if you could, we don't know what that window for normalization is in non-rodent species because the data's not there, okay? So here are the other big things. Germ-free mice have poor altered intestinal permeability. Their guts are leakier than normal mice. Their blood-brain barrier is also leakier than normal mice. So we're already, this should be sounding kind of bad, okay? They have changes in the receptors of various neurotransmitters in their brain and in their gut compared to regular mice, okay? So we know these things, and then we have to talk about, well, how does this affect it? So we know it affects their behavior in some ways. We also know that early stress has greater impact on the gut microbiome and these physiologic aspects. So stress that happens early in life has a greater impact than a similar stress that occurs later in life. So we're just going to look at serotonin for a second. I don't want you to think this is the only thing that's important in terms of neurotransmitters, because it's not. But serotonin is critical for the homeostasis in the body and the brain, and also in the GI tract. It's involved in a lot of functions. It's not just anxiety and aggression. You know, it's not just our little realm. It's fluid balance, intestinal motility, like it has a lot of effects, okay? So it is also a key neurotrophic factor in the fetal brain, meaning it helps build normal neurons, right? Having too much serotonin is bad, and having too little serotonin is bad. Everything's a balance. You know, everything in moderation to some degree, except for maybe wine and chocolate and cheese. So early life stress alters the serotonin transporter. So for those of you that don't know this or forgot, the serotonin transporter is a little receptor or protein that once serotonin is dumped into a synapse, that little transporter cleans it out. It takes it and brings it back into the cell, okay? And it does that so the serotonin doesn't hang out there too long or can sort of continue to accumulate, all right? So if you block the transporter, for example, with Prozac, or you damage the transporter by breeding special mice that have a dysfunctional transporter, then you alter serotonin function. You can make it have too much, for example. So in the intestinal tract, they breed these mice where they've got this screwed up serotonin transporter, and then they stress the mice. And usually it's maternal separation, because that's a big stressor for young individuals, obviously. So what they find is the mice that have the genetically modified transporter have more exaggerated changes under stress than the mice that have normal transporter function. So we know stress is a problem, but we also know that the serotonin transporter function is part of the deficit picture that we see in these germ-free animals or in animals that have transporter dysfunction for some other reason, okay? So we also know that even in adult individuals, there is downregulation of that transporter. That means it doesn't work as hard, or there's not as many of those transporters in situations of inflammation and diarrhea, particularly intestinal type things like Crohn's or inflammatory bowel disease, all right? And what this does is it reduces clearance of serotonin from the synapse, and then that excess serotonin leads to a variety of undesirable things, like there's something I just learned about called dumping syndrome, which apparently happens after you have like a gastric bypass. You know, they take part of your stomach out for obesity surgery, and then your serotonin system gets messed up, and then you get this dumping syndrome, which I don't know a lot about it, but it sounds bad. And as I mentioned before, those microbiota, certain types of them, can produce their own serotonin. So again, they help influence whether we have enough serotonin, too much, or too little, okay? All right, so what about probiotics? Because usually what they do with these studies is they say, okay, we make these germ-free mice, we stress them out, we do all this stuff, and then we see if we can fix it somehow, all right? Probiotics are live microorganisms, so they are actually live bacteria that you ingest to try to recolonize or normalize the diversity in your gastrointestinal tract, okay? Different species of animals have different concentrations and ratios of microorganisms in their microbiome, although there's a lot of overlap amongst the species, okay? So these different microorganisms also have different effects. So for example, lactobacillus species have anti-anxiety effects. If you take one of those mice and you give it a probiotic that has lactobacillus, and then you stress the mouse, the ones that have lactobacillus probiotics show less anxiety than the ones that do not. So for example, Lactobacillus reuteri increases plasma oxytocin. We all think about oxytocin as, you know, like the feel-good, bonding, mothering hormone, but it has a bajillion other effects as well. This organism also reduces pro-inflammatory cytokines, right? It also upregulates T cells, which is a type of an immune system cell that's good to have because they help fight infection and inflammation, right? Helveticus reduces peripheral stress hormones, so that corticosterone. They also increase anti-inflammatory cytokines, so this helps produce the good kinds of cytokines. And then Bifidobacterium longum is another one that's used a lot, and they found, again, when you give this, the corticosterone levels were reduced after stressful experiences, and it helped prevent those stress-induced changes in gut permeability. So then there's prebiotics, and prebiotics are non-digestible compounds that basically create changes in the microorganism that have beneficial effects on host physiology, okay? And this is usually by fermentation. So not only do prebiotics help sort of increase the population or allow for growth of good bacteria like Bifidobacterium and Lactobacillus, but it also helps, those products help those bacteria produce compounds that are beneficial to your health. And most of these are what we call short-chain fatty acids, right? So Bifidobacteria and Lactobacillus are two of the types of bacteria that are involved most in a lot of our pre- and probiotic studies, right? So here are some of the three most common short-chain fatty acids. So there's butyrate, acetate, and propionate. These are neurohormonal signaling molecules. So in and of themselves, they have direct effects on different physiologic processes. All right, here's one of those other kind of bad diagrams. But intestinal lining again, epithelial cells. Remember the little finger brush borders. So this is your lumen. This is the body side. Here are some of our lymphocytes down here. All right, so these are immune cells. Don't worry about that. You don't really need to worry about that. But these cells, again, produce some of these cytokines. So IL-6, IL-1, anywhere you see TNF, which is tumor necrosis factor, those are basically bad cytokines. IL-10 is an anti-inflammatory cytokine, so it's good. So what happens is these bacteria ferment those non-digestible carbohydrates in the prebiotic. They produce these short-chain fatty acids, and those short-chain fatty acids can be used by energy, or for energy by either our body, but sometimes they're also used as energy by the bacteria themselves. But those short-chain fatty acids also influence the cells to produce good cytokines or sometimes bad cytokines, but these are mostly the good kind of cytokines. These also can stimulate gut hormone release from the neuroendocrine cells, and these can pass through the bloodstream up into your brain and have direct effects on your brain. So we're going to look just a little bit about butyrate, because there's a lot of study done by that, and it seems to be sort of the most beneficial. That might just be my impression from my limited research. But butyrate has direct beneficial effects on brain plasticity and function. So that brain-derived neurotrophic factor I mentioned before, butyrate increases and enhances that in your hippocampus, and what you end up is an increased ability for what we call hippocampal learning. So the hippocampus is involved in memory formation and also spatial memory formation. So individuals that have higher butyrate levels, again from good gut bacteria, do better on the types of tests where they're assessing spatial memory, okay? Butyrate also has direct antidepressant effects. So we don't completely understand still some of all the specific, like we don't know exactly how butyrate has an anti-effective or antidepressive effect just yet, but we know that some of these effects exist. Like it says, this isn't a complete lecture for one thing, nor is all of this completely understood. But butyrate has been linked to improvements or changes in symptoms from a variety of physical and what we consider mental disorders, okay, like autism spectrum disorder, Alzheimer's, and anxiety and depression. So we also know that acetate, just as another example, can cross the blood-brain barrier and influence gene expression in your hypothalamus to have, again, various effects after that. So I'm not going to go through this really, but I just put a quick list up here. These are some of the more common organisms that are in our canine and feline GI tracts. These obviously change based on health or disease state, which I'll allude to a little bit, but I just kind of, again, wanted you to kind of have a list for whatever good that does you. So what if we look then at disease? So all of these conditions have been linked to the microbiome either a little or a lot. So for example, the microbiome effects on cognitive function are somewhat mild, but the microbiome effects on anxiety and depression are consistent and clear. The microbiome effects on obesity are consistent. Again, not completely understood, but there is a definite, definitive link between your biome health and weight maintenance. If you take germ-free mice, or even regular mice, if you basically take fat mice and you fecal transplant them with the microbiome from a skinny mouse, they will lose weight. If you take fat mice and you transplant them with the microbiome from a skinny person, they will lose weight, and vice versa. If you take a skinny mouse and give them the microbiome from an obese person, they will gain weight. So again, there's something going on with this connection and these hormone productions that affect a lot of gene expression in the body in a variety of ways. It's not just like, oh, it makes me hungry. It's much, much more complex than that. We spoke a little bit already about atopy or allergies and food intolerance with the hygiene hypothesis. I'm not going to go into autism spectrum disorder other than to say part of the issue there is apparently a lot of individuals that have ASD also have pretty significant gastrointestinal symptoms, and there is, again, fluctuation related to their GI health and their autistic, or some of their autism-related symptoms, but I'm no expert on that, so don't really ask me about it because I can't answer. So let's get back to stress again. We talked a little bit about early life stress, and that can have profound and permanent influences on your microbiome and your stress response system, okay? And we've known that for a long time. If any of you ever followed some of the grooming studies in rats where they showed if the mothers were stressed, it changed how they groomed them, and then it affected the behavior of the pups actually across generations. Some of those studies are now making a little bit more sense to me because the microbiome's probably involved in a lot of that, all right? But repeated or chronic stress alters your GI microflora. And it does it in a variety of ways, and these aren't all completely understood. But again, it's bidirectional, meaning sometimes it's the brain sending certain signals down that increase the inflammation in the GI tract, and obviously sometimes things are happening in the body or the GI tract that are sending signals back to the brain, and this sort of becomes a vicious type circle, right? So we know chronic stress increases intestinal permeability. The microbiota are involved in the regulation of the proteins that form those tight junctions. So if you mess up the good microbiota, and they can't do their job, those tight junctions start to fail, and then you get breaches in the intestinal barrier that allow a lot of that junk in your GI tract, which the body's trying to keep out, that stuff now can start to move into the body. That includes translocation of the bacteria themselves, all right? Meaning you breach that barrier, and if you're harboring some E. coli or salmonella or something in your GI tract, and you get a leaky gut, and those bacteria breach that barrier, now you have a systemic infection. Your systemic infection is going to produce a lot of those cytokines, which will either augment or kill, if you will, the good and bad bacteria. And then you can kind of see how this starts to cycle. More inflammation in the body means more breaches in the intestinal permeability, also causes changes in the permeability of the blood-brain barrier. So can you kind of envision, even though we don't understand all of this, how this turns into a giant mess, okay? The other thing that we know, so CRF is corticotrophin releasing factor. This is the hormone that the hypothalamus secretes to stimulate the HPA axis. So you know, hypothalamus, pituitary, adrenal axis, each one of those organs, HPA, secrete a different hormone. And CRF comes out of the hypothalamus, and it releases, causes the pituitary to release ACTH, which, by the way, has primary anxiogenic properties, as does this. But local release of CRF right around the cells rather than into the bloodstream that goes out to the body, local release can lead to changes in the blood-brain barrier permeability as well. So not only does the CRF in the bloodstream create changes in the intestinal and blood-brain barrier permeability, but the sort of stuff that's just leaking locally can influence permeability of the blood-brain barrier also. All right, you still with me? Okay. So, increased HPA axis alters intestinal permeability. Not sure if it is because of direct actions, say, of the corticosterone itself, or is it because it influences action of the vagus nerve? Again, that kind of stuff I don't think is well teased out yet, but this is not in contention. When your stress response system kicks in, intestinal permeability suffers. So for those of you that work around dogs, especially in the veterinary profession, how often have you seen stress colitis? Okay, there you go. This is why it happens. So frankly, every dog and cat in boarding should be on what? Pre and probiotics. That's my opinion anyway. So how do the probiotics help us with that? Well, they can help reduce some of these stress-induced changes. So again, Helveticus, Bifidobacterium longum, they help restore tight junction integrity because those little proteins, they help affect the gene transcription for producing those proteins. So if we have the good bacteria to help the body produce the right proteins, then we maintain tight junctions. But if these ratios are messed up, basically, and we don't have enough of these bacteria, then we lose integrity of the tight junctions, and I'm sure this is quite simplified, so, you know, bear with me. I'm sure it's much more complex than this, but this is all I know about it. Then again, we lose the integrity there, and then bad stuff comes through, okay? These probiotics also influence adherence and translocation of pathogenic bacteria. So one way bacteria get in, you know, most of them are free-floating in the lumen, and remember I said there were two layers. There were mucosal bacteria, and there were luminal bacteria. So if the luminal bacteria get in, and they're bad ones, let's say, and they get into the mucosal layer, then they can sort of stick and either influence these cells or maybe pass through those big gaps, and then you got food poisoning or whatever, you know? So these good bacteria help protect not only the tight junctions, but help protect against translocation, as I said, and adherence. So what about depression and anxiety? These are clearly linked to function of the microbiome. Like I said, some of the other stuff is a little bit inconsistent and sketchy. This is consistent. Depressed individuals have a different gut composition than normal individuals. And individuals basically that have systemic or intestinal inflammation are often more depressed or anxious than normal individuals. And it's a chicken and egg thing, okay? So it might start one way. So depressed individuals sometimes have an aberrant HPA axis regulation, so their stress response system's already screwed up. They may have chronically elevated cortisol levels, and we already know that HPA axis function can be detrimental to the intestinal barrier function, right? So if you're depressed, you weaken your GI tract, and then, again, bad stuff happens. Or if you have inflammatory bowel disease, that goes back to your brain, all that chemicals and stuff, and makes you depressed. That's linked, okay? Presence of pathogenic bacteria, anything that creates systemic inflammation can create depressed or anxious-like behavior, all right? And remember all those cytokines I talked about? So some of those cytokines have direct effects, again, on your body and your brain. If you take individuals that have inflammation and you give them anti-inflammatories, they show less anxiety or depression. That's why there's this little trend in human medicine, again, about the link between depression and inflammation. Are some of these people depressed because they have inflammation? And sometimes treating them with anti-inflammatories improves their depression. That doesn't mean we all need to go out and like start chugging ibuprofen, but the link is there. The other thing that they've found is in people that have depression, some of the metabolites that they find in those people are direct byproducts of the gut microbiota. Like the gut bacteria made those products. They get into the systemic system, and then you have depression, okay? Again, it's more complex than that, but the link is there. Depressed people have higher inflammatory cytokines. So again, the bad ones, IL-1A, IL-6, tumor necrosis factor. And again, if you inject normal animals with these cytokines, you will make them depressed or anxious. And if you take individuals, like if you take a mouse and you give it an infection, it gets anxious and depressed, and then you clear the infection or you give them anti-inflammatories, they behave more normally. So again, this is not in dispute. This connection is very clear, even though we don't understand all the mechanics behind it yet. So what about irritable bowel syndrome and inflammatory bowel disease? This is where most of the study with the biome has been done in animals, like our animals, dogs and cats, horses, stuff like that, but also in people. So people that have Crohn's disease, IBS, IBD, frequently have more depression or anxiety. And again, is it a chicken or egg? I don't know, that's not my thing. They also often have changes in their attention, their emotional regulation, and their memory and cognition. We do know that humans and animals with IBD have different ratios of their gut flora than normal animals. That is established in dogs, cats, horses, and people, and rodents. And I'm sure there are other species, but I didn't go into any bizarre ones like rabbits and squirrels and things like that. So generally, there's lower numbers of bacteria, and there's less diversity. Sometimes there's also obviously ratio shifts of what normally is the predominant organism is now not the predominant organism. So diet, huge impact on your microbiome, okay? You are what you eat, which is why I wrote for those of you that wrote the haiku, I'm sorry, I actually wrote the haiku that says, we are shit for brains, because it's true, okay? So we know, because everybody pounds this into us, that a healthy diet is a reduced risk of lots of health issues, but it also reduces your risk for cognitive problems, anxiety, and depression. And now we have a little bit of a feeling for why, all right? So if you take animals, for example, mice, and you give them a crappy diet to the mothers, i.e., high fat, low fiber, because remember, mice are kind of like horses. They're hindgut fermenters, so they need a lot of forage or roughage, and they don't eat high protein, high fat diets naturally. So high fat, low fiber diets are abnormal for rodents. So then you get less microbial diversity in the offspring. You get increased colonic inflammation, because again, we're going to change the barrier permeability. And then there are intergenerational effects. After 4 generations of this, you can't fix it. And I don't have kids, but I just started thinking about that, and I was like, wow, how depressing is that to think if your ancestors ate crappy food for generations, that you're now messed up and you might not be able to fix that. I don't know, it just came to me. So, sorry those of you that have kids, go home and eat better. No. So this is the Western style diet. You know, everybody says Western, you know, we eat a lot of processed foods, a lot of sugar, and a lot of fat. And I took a webinar a few years ago where they talked about sugar, and honestly it was shocking, because they were like, back in the, I don't know, it was like maybe the 1700s or whatever, the average Westerner ingested like, it might have been even earlier, maybe it was the 1600s or whatever, the average Westerner ingested like 2 tablespoons of sugar a year. And now, what do you think the number is now? So now, so that was 2 tablespoons a year. Now it's 200 pounds per year. Yeah, that's what I did. I was like, oh! But I'm guilty, and then I still pour sugar on my, you know, Raisin Bran in the morning, and I'm like, I shouldn't be doing this. Those damn endorphins and dopamine. So, high protein, high fat, low fiber diet. Basically, that's what's going to kill you. Literally, it's going to kill you, okay? Well, in conjunction with high fat, low fiber, yes. It's not the protein itself. This is just what Westerners tend to eat. You know, like we love our meat, not me so much, but I mean, really, you know. We eat tons of meat, whereas the Mediterranean style diet is low inflammation diet, which is why people that have IBD and things like that, diabetes, this is what they want you to eat, because it is high in legumes, so beans, okay, chickpeas and things like that, those are very high in prebiotics. So that means when you eat those foods, you are growing good bacteria, and you're giving those bacteria the fuel they need to produce short-chain fatty acids and influence cytokine production in the right direction, okay? So reduced risk of depression and obesity, and as I said, increased production of short-chain fatty acids. So then we say, okay, well, this is an animal behavior conference. What about our animals? Well, this is pretty much what you're going to see in every slide. There are no studies evaluating behavior in the microbiome in dogs. There are, however, studies looking at inflammatory bowel disease in dogs and the gut microbiome. All right? There's several of those out there. And just like I mentioned before, the bacterial diversity is lower, the ratios are shifted, and the microbial numbers are lower than normal animals. Okay? So there are different metabolites, and we see lower levels of Firmicutes, Bacteroideae, and Clostridia. These are usually pretty populous in the healthy canine GI tract. All right? So there is a study that came out in 2017, brand new study done by Purina. And what they did is they looked at microbiome changes in dogs that were on moderate protein, high carb, and then high protein, low carb. So 48% protein, 10% carb. I don't remember the carb source or the protein source. Maybe it doesn't matter. Versus 25% protein and 38% carbs, which is pretty much like our average dog food, right? And what they found was that they took fat dogs, labs and beagles, and they took normal weight, yeah, right, how hard is that? And they took normal weight dogs, and the changes in the microbiome between the dogs was more pronounced in the fat dogs. And those dogs had a microbiome on the high protein diet that was more reflective of weight maintenance gene production. Okay, so basically, just like they found in cats in the past, and if Dr. Case is in here, if I say anything wrong about diet, correct me, because I'm not a nutrition expert. The high protein, low carb diets promoted a more healthy gene expression for weight maintenance and loss. All right? All right, so here we go. No behavioral studies. This is the one that threw me. And again, Dr. Case in here somewhere? I saw her back there before. Nope, yeah. She might have something to add to this. So they took cats, fed them high protein, low carb diet. So 53% protein, and they didn't list the carbohydrate level, so I don't know what it was. And then a moderate protein and moderate carbohydrate diet. What they found was that the cats that were on the high protein diet had lower levels of the good bacteria. So then I'm like, because everybody has been telling me when I talk to nutrition people and feline people, we need Atkins cats, you know, we need high protein, high fat, low carb to keep our cats healthy. Then this study comes out, and they did a similar one in kittens. This study comes out and says that these cats on the high protein diet had a gut microbiome that was more suggestive of cats that have inflammatory bowel disease. I don't know what to do with that piece of information. It is what it is. So hopefully people that are smarter than I will follow up on that and then tell us where to go, because right now I just parrot. I'm like, yeah, low carb diets for cats. Don't know. So here are the kittens fed high protein, low carb. They had lower numbers, typo, again, of bifidobacterium compared to cats on medium protein, medium carb diet. So again, bifidobacterium is associated weight control and weight loss and is sort of negatively correlated with inflammatory bowel disease. So I don't know what to do with this piece of information either, because this seems to go counter to what we know about cat nutrition so far. And do you have anything to add to that? Quality of the protein, she said? Okay.
Who sponsored the study?
I'm sorry?
Who sponsored the study?
I don't know that this was sponsored. I know the other one was done by Purina. I don't know that this was sponsored by anything. Okay. All right. So horses. No behavioral studies. What we do know is colitis, non-specific colitis or undifferentiated colitis, they call it, is associated with lower numbers of Firmicutes bacteria and more, ironically, bacteroids and proteobacteria. And this I found shocking. 100% of horses that had less than a 50% profile of Firmicutes will develop postpartum colic. Like, that's pretty shocking, you know? So that means if you're breeding mares, man, you better get their diet right. Get them on probiotics and prebiotics. We know that NSAIDs reduce microbial diversity in horses. So obviously certain antibiotics affect our GI tract function too, and this is well known in the hindgut fermenters. So those of you that work with rabbits and hamsters and horses, you know, antibiotic therapy is like a big scary thing because sometimes it changes those ratios enough and those animals can literally die from that. Okay. So the other thing to think about is changes in gut diversity, of course, influence systemic inflammation, right? So we talked about that. Everybody still on board with that? Okay. How many horse people are in here? All right. Laminitis. Laminitis is a systemic inflammatory condition in horses that affects their feet. It can absolutely be life-threatening because you can't really like lop a horse's leg off and put a prosthetic on it. It's been done, but you know, it's not really practical in a 1,000, 1,200, 1,500 pound animal. So those changes in gut diversity, if they lower those short chain fatty acids and lower our butyrate and stuff, and then we get reductions in good cytokines, increases in bad cytokines, you get systemic infection, and not only could it influence laminitis, but it also influences insulin resistance. So insulin resistance is a big problem actually in a lot of our Western animals. It's not just people, but type 1 and especially type 2 diabetes in people is related to insulin resistance, fat cats, diabetic dogs, and insulin resistance in horses. Parrot people, I'm sorry, there ain't squat. Actually, I take that back. There are actually a few studies on, I don't know how to pronounce this, the kakapo. Is that right for you parrot people in here? No? Okay, well, I can say it wrong then. So this is a kakapo. It is an endangered, flightless New Zealand parrot. It is the only parrot that I found anything about the microbiome on, and it obviously didn't have anything to do with behavior. It probably has to do with conservation issues and health. But there are actually a few studies looking at microbiome effects in the kakapo, but that's all I got to offer the bird people. Sorry. The birds are always left behind. All right, so this is Charlie. He's one of my cases. I've been working with Charlie for a really long time. And Charlie was found in a meth house when he was a puppy. He was adopted. The owner, who is fantastic, loves this dog more than oxygen, which she will freely tell you. She is fortunately, you know, affluent enough to cater to all of his needs. And so she brought the dog in, and he was a mess. He's aggressive to dogs and freaked out and scared and aggressive to people, and you couldn't get him into the veterinary. And he was totally unruly at home. Like, she came in with her husband's shirts, and they had like holes ripped in them because the dog would jump up and grab him and then literally rip the shirts right off of his body. He was kind of a mess, all right? Body handling problems and a lot of generalized anxiety. So Charlie also had chronic intermittent diarrhea. He had food allergy, and he has atopy, all right? I'm sorry? Atopy are pollen allergies, basically, like inhaled allergies, grasses, stuff like that. So we got Charlie stabilized on sertraline, trazodone, Hill's ZD, which is a hydrolyzed protein diet, and hyposensitization injections, which were a chore to get into the dog, I will tell you. And Charlie is actually doing really well. I mean, his diarrhea is pretty much gone, the itching controlled, as long as he didn't get anything. She's super religious about keeping all contraband foodstuffs away from him and things like that. So Charlie's been stabilized for probably 2 or 3 years. His behavior's in a good place. We just continue to work with him, especially on husbandry stuff. But really, he's in a good place, stable. So I know this dog well. So then, Charlie's put on pentoxifylline by his regular vet, who happens to work in my practice, for ear margin vasculitis, all right? And Charlie breaks with diarrhea that we cannot get under control. Like, the dog had diarrhea for 9 months, and it's not because we suck at our job. And so we went through all kinds, you know, he was on metronidazole, we gave him cobalamin injections, we did GI panels on him, we checked his fecals, we put him on Tylenol, I put him on probiotics. And finally, after 9 months, we added in high fiber and maybe whatever, and Charlie is back to good. However, for those 9 months, I completely lost control of his behavior. Like, the dog, not completely relapsed. His anxiety level was probably 40% higher than his baseline. And I'm like, I don't know what's going on. You know, maybe because he's got diarrhea, he's not absorbing his medications right. We started playing around with his medications for behavior. You know, I'm like, all right, well, let's quit that protocol and switch to something else. Because back then, I mean, I knew probiotics were good, but I didn't really understand why, and I didn't understand all this data about the microbiome. But, you know, now this makes complete sense to me. And when we finally got the dog's diarrhea back under control, his behavior improved again, and not a cause and effect. So, Dr. Case, I listened to your lecture yesterday. But since then, what I think is interesting, and again, maybe it's not a cause and effect thing, he no longer needs a hypoallergenic diet. So he's on a regular dog food, and he's allowed to have other kinds of treats and stuff like that, and he doesn't break with diarrhea, and he doesn't itch anymore. And I don't know, I mean, like, again, I'm not trying to say that, oh, we fixed his microbiome, and now we fixed everything, but I do think it's interesting, because I knew this dog's history for a long time, like a long time, and I knew what set him off and what didn't, okay? But I have a lot of cases like this. Like, I really am sort of like a secondary gastroenterologist, because I cannot tell you how many behavior cases I have that have chronic gastrointestinal problems, and we absolutely, in many of them, not all of them, can journal waxing and waning of their GI symptoms and their behavior. Like, I had a golden doodle owner that had resource guarding and owner-directed aggression, shocker, and literally, and he had food allergy and chronic intermittent diarrhea, you know, and literally, as we're working on that, if he started to get punky off of his food, like, literally they knew. They go, within the next 24 hours, he's going to try to bite us. Like, they absolutely could predict it, all right? So I see this a lot, and I think this is partly why I got so interested in this topic, is because I see it clinically so much. So what we do know, microbiome disturbances are going to affect behavior. Not every behavior, and it may not always be clinically relevant, but there's enough data there that we need to pay attention to this, okay? So how's that going to influence your behavior modification? So when those animals have GI upset and they don't eat, you know, it affects our treat training, it affects their potentially memory and cognition, not only because they feel bad, but because the inflammatory condition that's going on in their body, or again, the changes in their microbiota that is affecting their brain and behavior. Perinatal effects can last a long time and forever. And when I was going through my residency, and I had to read a bajillion textbooks, many of which y'all have read, and how many of you read in references that say puppies that had parvo or puppies that had health issues when they're young are more likely to grow up with behavior problems? Maybe this is why. Maybe because it's an early life stressor, while their HPA axis is still sort of developing, that they got screwed up, and that's going to be a permanent reflection in their physiology. Because it's not like we can scrape their microbiome out and go, here, have a new one, But for me, what this means is I would be a lot more aggressive now about treating or thinking about the microbiome in perinatal situations in ill neonates and pediatrics. That's me. That's partly what I take out of this. There are transgenerational epigenetic effects. All right? What happens to the mother affects the kids, and if those kids have kids, it passes on. Sometimes you can fix it before they have the grandkids, but sometimes even if you try to fix it, it's just like those changes in HPA system in the grooming rats that is transgenerational, okay? Basically permanent. Diet's critical, so we know what people need to eat. We know what horses need to eat. I think we're still trying to figure out what our dogs and cats need to eat or what we can feed them, because it's not like we're out catching mice for our cats, which maybe would be smart, but it's not going to happen. So if we're going to formulate a commercial diet or a homemade diet, I think, and again, please jump in, Dr. Case, that we need more info here, because we know a lot about nutrients and we're learning more about protein effects, but I don't know that any of that's yet been tied into the effects on the microbiome. And then I see these few studies that are a little bit contradictory for me, so I don't know. I don't have any advice for you about diet, but maybe in a year or 5 years we'll have more data where we can blend all this information together and make smarter choices about what we feed our dogs and cats. There is some data to show that antibiotic therapy, especially in certain types of colitis, so it's kind of our knee-jerk reaction in the veterinary world, that when dogs get colitis, that we stick them on metronidazole, and there is actually some data that may actually hinder recovery from diarrhea and colitis. So that wasn't done in dogs, it was done in some other species, the rodent models, but that may not influence you, but I do think we have to be careful, and we try to be careful more and more about antibiotics. So for example, that dogs and cats maybe that have skin infections, instead of just plugging them with oral antibiotics, maybe we need to try topical therapy first so we don't screw with their biome unnecessarily. So pre and probiotics, like as soon as I started doing this research years ago, like I take probiotics all the time now. Like to me, there's just like no reason not to, and I probiotic everything. I probably don't do the cats as much as I should, but because I see so many dogs with GI issues, I literally now almost probiotic everything that comes into my office if it's a dog. So I'm definitely going to do it if there's any GI symptom. If they fart a lot, probiotic. I'm serious. If they eat grass and puke periodically, probiotic. Like, they're getting it. Because unless you have a serious issue with leaky gut, like your intestinal barrier is basically destroyed, these are basically side effect free. But if your intestinal barrier is completely wrecked, there's this smidgen of risk of dumping live bacteria into something that's that dysfunctional, okay, even if they're good bacteria, all right? You're not going to see those. But I also hope that we're going to see someday, and there'll be work on this, are individual probiotic therapies based on age, all right? So for example, and species. So cats have a higher level of Enterococcus in their microbiome than dogs do. So when I learned that, I'm like, aha, that makes more sense to me then to maybe lean toward something like Fortiflora for cats, because Fortiflora is Enterococcus, all right? But for dogs, I use ProViable, and there are other goods, but the reason I use this product versus Fortiflora is because, again, when I started doing this research, Fortiflora has one microorganism in its Enterococcus. ProViable has 7, and it emphasizes the Lactobacillus and Bifidobacteria species, which seem to be more important in the research that I'm reading. So for me, this is what I reach for my canine patients. But for cats, especially maybe kittens, maybe Fortiflora is good or better or whatever. But hopefully we will see enough data in the future where we can tailor this more instead of a one-size-fits-all kind of approach, which is what we do now. There are also probably going to be maybe individualization based on the condition that you have or the individual has. So those are sort of my dreams about pre- and probiotic supplement in the future. And that's all I have. Almost right on time.